My name is Ian Brown, and my work is in the Chemical Engineering Department under Dr. Ryan Summers, with my project specifically being on characterizing the behavior of the twister sister ribozyme for the development of synthetic riboswitches. Now, to begin, I'd like to explain what a riboswitch is and help provide a visual of how it actually functions. Put simply, a riboswitch is an mRNA fragment which regulates gene expression through conformational changes. To help visualize how this works, I'd like to direct your attention to the top diagram, labeled riboswitch off. First and foremost, that blue box labeled RBS stands for ribosome binding site. And as you can see, the blue oval labeled ribosome is actually unable to access said RBS. This is due to the presence of the twister sister ribozyme, the shape labeled twister sister. And due to the shape of the twister sister ribozyme, the RBS is actually hidden from the ribosome. The result of this is that the GFP gene, represented in green, is actually not expressed. Compare this to the second diagram labeled riboswitch on. In this case, due to a change in the shape of the twister sister ribozyme, the RBS is actually accessible to said ribosome, and the result of this is that the GFP gene is expressed. Now, the two things I'd like to highlight here is that the self-cleavage of the twister sister ribozyme is actually responsible for the shape change and corresponding expression of the GFP gene. I'd also like to point out that the twister sister ribozyme is serving as part of the mRNA fragment that I'm referring to. The application of these riboswitches is actually a powerful tool for the control of gene expression. From this example, we're controlling the expression of the GFP gene, but you can control a variety of genes, both in research and industry, especially when you're looking to produce certain protein products efficiently. Next, I'd like to talk about the twister sister ribozyme itself. This is actually imaged there on the right half of the diagram. It is a novel class of ribozyme that is actually similar in structure to the previously characterized twister ribozyme. This is represented on the left half of that image. Now, the twister ribozyme was previously discovered to be self-cleaving, and thus it is suspected that the twister sister ribozyme will also be self-cleaving. One more aspect of this diagram I'd like to point out is the distinct segments of the twister sister ribozyme. These are P1, P3, P4, and P5. Now, put simply, these just refer to where the twister sister ribozyme will actually be implemented into the mRNA fragment. But I will be referring to these segments as P1, P3, P4, and P5 for the remainder of this presentation. The large question then, is the twister sister ribozyme self-cleaving in vivo, and can it function as a riboswitch? To test that, we move to the in vivo fluorescence test. Now, to accomplish this, the GFP gene was actually implemented into E. coli cells, and you can see that from the plasmid map visualized there on the left. And if you look in the red circle, you can see in this example, the P1 ribozyme is implemented into the plasmid just before the GFP gene. The expression of this GFP gene is controlled by the twister sister ribozyme, with self-cleaving behavior allowing for the expression of the GFP gene, which will actually produce the fluorescence that you can see in the image on the right. Now, with the picture on the right, the plus refers to the positive control, where the GFP gene is known to be expressed, and as you can see, we do in fact have fluorescence. The negative control, we do not have fluorescence. However, when looking at the P1 tube, you can see that there is no fluorescence, which actually leads to suggest that the twister sister ribozyme is not self-cleaving in vivo. And similar results were actually found for P3, P4, and P5. Now, this result actually led us to move to an in vitro test to confirm any self-cleaving behavior of the twister sister ribozyme. This is where the majority of my work resided, and this test was completed in 3 main steps. First and foremost, the transcription of the twister sister ribozyme. Next, each ribozyme fragment was run on a denaturing page gel. And finally, we confirmed our results by comparing the RNA bands present on the gel. The first step of this process was transcription. To do this, a DNA template that was previously linearized and amplified by PCR was used. And I'll help explain what this looks like in a moment. The ribozyme product was produced and its concentration was measured. Importantly, this is a single-stranded RNA product, which will also be visualized in a moment, and it is the P1, P3, P4, and P5 fragments I mentioned earlier. The final note I want to point out is that due to the presence of magnesium in the transcription product, our ribozyme product will show any cleaving behavior upon the completion of transcription. Now, put simply, this means that if cleavage occurs, our product will actually consist of two differently sized RNA bands from the single mRNA fragment that was cleaved. And this is supported by the graph there on the right. And simplified, as the increased concentration of magnesium, represented by the x-axis, actually leads to an increased cleaving behavior, represented by the y-axis. Now, this diagram here gives a very general and basic look at what is actually happening in our transcription of the ribozyme. The DNA template, represented in blue, is actually separated, which allows RNA polymerase to interact and produce the mRNA fragment. This is the P1, P3, P4, and P5 fragments I was referring to. And as you can see, it is a single-stranded product produced from our DNA template. The next step of this process is running these fragments on the denaturing page gel. What this is polyacrylamide gel electrophoresis, which is simply a technique to separate nucleic acids based on their size using electrophoretic mobility. To do this, a charge is actually applied to the gel, and smaller fragments are actually going to migrate farther down the gel than larger fragments, which will not migrate as far. This will actually separate the fragments based on their size. The denaturing portion of this term simply refers to linearizing the RNA, which helps better measure its size. And as I mentioned before, the presence of two distinct bands will actually confirm the twister ribozyme showing self-cleaving behavior in vitro. Next, let's move on to the confirmation of our results. First, I'd like to draw your attention to the red arrow labeled P1. Now, this is the P1 fragment that we tested, and I'd like to point out the two distinct bands that you can see on the gel. As I mentioned before, these two distinct bands were actually compared to the standards, represented there in purple, to confirm that we were actually getting the sizes we were looking for. And as I mentioned, the two distinct bands actually does confirm that the twister sister ribozyme is self-cleaving in our transcription process. And as you can see from the P3, P4, and P5, represented by the arrows to the right, the blue, orange, and green arrows, they also have two distinct bands, showing that all fragments of our ribozyme were in fact cleaved. I'd also like to address some of the extra bands in this, some of the background. This simply refers to unreacted template DNA or uncut RNA that we couldn't actually purify out of our product. And this is due to the extremely small size of the fragments we're working with. If we did attempt to purify, which we did before, these mRNA fragments would actually be lost due to their extremely small size in the purification process. Now, as I mentioned, the big conclusion from this is that the twister sister ribozyme does in fact express self-cleaving behavior during transcription. Now, where does this lead for future work? Well, first, we can attempt to test the functionality of our ribose switch in vitro. To do this, we could attach what's known as a ligand-binding aptamer to our ribozyme. And what this essentially allows for is better control of gene expression and the actual cleaving behavior of our ribozyme with the presence of a ligand. In our case, we'll be working with theophylline. We can also move to in vivo self-cleaving experimentation like we did before. We can begin to re-examine our previous in vivo experimental protocol, as from our in vitro test confirming self-cleaving behavior, we should expect to see self-cleaving behavior in vivo as well. And we also can begin to differentiate between cleavage and activity, as currently we're using the expression of the GFP gene to confirm any cleavage. If, however, there's a problem in our experimental design where we are getting cleavage and not expression, we currently are not able to differentiate between those two factors, and that's something we would like to be able to do. Finally, I'd like to highlight the ultimate goal of our project, the completion of a fully functional synthetic ribose switch for the in vivo control of both gene transcription and translation. Thank you for listening. I'd like to thank Dr. Ryan Summers, Xuyan Zhang, a graduate student on the project, and Ifham Hawk, a previous undergrad, as well as the NSF for funding. Thank you, and do you have any questions?
Could you explain in a little more depth about what differentiating between cleavage and activity means?
Yes. So as of this current moment, what we're actually using to determine if our ribozyme is self-cleaving in vivo is the expression of the GFP gene. As I mentioned earlier, if we're not getting that self-cleavage, we're actually not having the shape change which allows that RBS to be accessed. However, if for some reason that we're not able to see right now, we are in fact having cleavage of our ribozyme, but our GFP gene is still not being expressed, we currently would not be able to tell, as we're only looking at the fluorescence to indicate a successful self-cleaving of our ribozyme in vivo.
Can you explain how the standards work on the cleavage gel?
Yeah, so the standards there represented in purple are simply used to actually determine the specific sizes of the bands we're working with. If we didn't have standards, we could tell that there is two distinct bands, but we wouldn't actually be able to tell the sizes of those bands. So what the standards are actually made up of is pre-known sizes of fragments. So I don't actually have the scale attached here, but there is actually a scale to tell you exactly the size of each band and how far it traveled. So for example, the two bands here, you could compare them to the bands on the standards, and if, for example, maybe 100 kilobase pairs, if it is lower, you would know that it is larger, and if it's higher, you know it's less. And from there, you can actually determine the exact sizes of the fragments on your gel.
Could you explain a bit more detail between the positive and negative control tests for your in vivo stuff?
Yes. So looking here at the positive and negative control, these were extremely critical in this test. As I mentioned before, the positive control, we need to actually visualize what the GFP gene, the fluorescence it causes when it is expressed, what that actually looks like as a comparison to our ribozyme, our P1 vial here. The negative control is the opposite. When we know the GFP gene is not expressed, we need to make sure we're not having any background fluorescence or anything like that. And because of that, we're actually able to compare if we are having expression of the GFP gene simply by looking at the fluorescence. And this makes things a lot easier when you can simply just visualize the gene being expressed due to its fluorescence or lack thereof.
Good job, Ian. Thank you.